Good morning and good afternoon, wherever you are. And thank you very much for joining this webinar, where I will try to tell you the molecular tale of biopolymers that is narrated by the size exclusion chromatography that we coupled with advanced detection. Well, when you first see this slide, I'm sure that you are thinking that you've been promised the story about the polymers and why you should actually care about history of medicine. Well, to begin with, it is fascinating and it's very fun because it is full of trials and errors, villains and heroes, but also it grounds us. It gives us a much better understanding of the world in which we live right now and tell us how we arrived to this point of time. It makes us appreciate much more the scientific advancement, because if we go back to the early ages of medicine and surgery, we can see that the surgeons were before using the metallic staples or even glue to close the wounds and stop the bleeding. However, this often led to a much more pain and less of a healing for a patient. And if we fast forward to today, we can see that the surgeons right now are using more biodegradable polymeric stitches or even medical tapes that not only hold the skin together, but they can as well deliver or release some active ingredients such as antibiotics or growth factors that will improve the healing process. Another example is the use of prosthetic masks that we moved from using them in the World War I for covering the injuries to the use of artificial organs and tissues that we can use to replace the diseased and injured tissues. And the last but not least, we also have to appreciate how we developed not only in the treatment of diseases, but also into the prevention, because just only 100 years ago, we had a population who was losing their life because of the simple disease. And right now we have many vaccines that can prevent them. And the most recent and most known vaccine is, of course, the COVID-19 vaccine that was developed in a record time. So this improvement of medicine throughout the history has been partially possible due to the development of the biopolymers. These biopolymers are macromolecules that have a very unique characteristics that allow them to be tailored for a specific medical function. For example, as I mentioned, the regenerative medicine, or even as a bioimaging, or as a drug delivery systems in the nanomedicine field. And in the last decade, the use of these biopolymers in nanomedicine has increased tremendously. And today I will guide you through the polymer therapeutics field, which is a subgroup of nanomedicine where we can use biopolymers as a drug delivery systems. As I mentioned before, we already know about nanomedicine and biopolymers have been used for the drug delivery systems. However, we are not so happy with the amount of these systems in the clinics. Why is that? Because mostly this journey from bench to bedside is a very expensive and a complex process, especially for the nanomedicine. And one of the reasons that we found that influence this clinical translation of these systems is, apart from the general lack of protocols or even the often inaccurate study design, it is also inadequate characterization. To tackle that, we need to use a fit-for-purpose analytical techniques. And even though that, as you can see here on this diagram, there are many parameters that we can characterize with our analytical techniques for these specific systems, today, since the amount of time is not enough, I will just cover of molecular weight, size, and the structure parameters that we can characterize by using size exclusion chromatography. What is size exclusion chromatography? As the name itself says, the size exclusion chromatography is based on the separation by size, meaning that the big and large molecules will elute the first from the columns, and the small molecules will elute later because they are small enough to enter the pores of the column and stay inside longer than the bigger particles. What is all of the components of the one instrument, size exclusion chromatography instrument, and their setup? So from the beginning, if we look at the SEC instrument, we do have a solvent or a mobile phase that we are using for the whole flow through the instrument. Then we have a degasser, which is used for removing the bubbles from the system, the pump, which is leading the whole mobile phase, and injection loop, which is used for the injection, as the name itself says, of our samples in the SEC column, where the complete size exclusion chromatography and the separation is performed. After the separation from these columns, the samples are entering the detectors that can be different. We will see in the future, in the next slides, which ones exactly. And finally, everything goes to waste. So what is the first thing that you have to do when you are developing a size exclusion method development, or when you get the sample? What is the first things that you need to take into account? I divided them into 4 different parts, and we're going to go through all of these, every single part separately, and discuss in details. The first part that we are going to talk about is the eluent or the mobile phase choice for your experiment. We can separate the mobile phases that we can use in the 2 big groups, the organic solvents and the aqueous solvents. And as you can see in here, the organic ones can also be separated into 3 subgroups. Why? Because they are different solvents that can be used either at the ambient temperature, a little bit elevated temperature, such as toluene, or even if you, for one of the samples, you need to use a really high temperature, set up to 210 degrees, you can use the trichlorobenzene solvent. And in the case of the aqueous solvents, you can have water and a different combination of the buffers for a different pH, but as well the buffers in the combination with methanol or even ethanol. So how are we going to choose the solvent for our analysis? Well, there are a couple of criteria that you have to think about. The first of them being that this solvent needs to completely dissolve your sample. Then the next, it must permit adequate detection of your analyte in the eluent, meaning that in the case of refractive index detector, the refractive index of your solvent has to be significantly different compared to the sample refractive index. And when it comes to the UV detector, the solvent needs to transmit this energy of the laser that is coming to your analyte. Then the solvent must not degrade the sample during the dissolution and the use, and during the analysis, of course, in your instrument. And the last but not least, it must not corrode any of the components of the chromatographic system. As I mentioned, refractive index is one of the parameters that needs to be considered when you choose that solvent choice and solvent. And one of the example is the PDMS polymer with a refractive index of 1.407. We can dissolve this polymer in 3 different solvents, in the THF, in the toluene, and the chloroform. Since the THF is the most common solvent, we first, of course, we're going to try to analyze the polymer with the THF. And as you can see here on the first line, well, there is no sample peak in the chromatogram. And if we look at the refractive index of the THF, which is 1.407, there is a difference of only 0.001 in the refractive index of our sample, which is then, of course, logical that we will not see the peak of our sample. However, then if we analyze these PDMS polymer in toluene or chloroform that have 1.443 refractive index, which is significantly higher difference than 0.05, we will see the peak of our sample in the chromatogram. While in the case of the UV detector, when choosing the solvent, we need to know the UV cutoff of each solvent, meaning that, for example, if we can look at the toluene or chloroform that have the UV cutoff of 245 nanometers, it means that we can use chloroform as a solvent for the analysis of our samples only if the analyte absorbs in the UV spectrum higher than the 245 nanometers and not below. So for each either organic or aqueous solvent, you can find here the different UV cutoffs and decide which solvent is appropriate for your analysis. Well, as comparison of the size exclusion chromatography with the typical HPLC systems, as I mentioned at the beginning, in the principle of the size exclusion chromatography, we want to separate our samples only by size. So we do not want to have any interaction of our samples with the size exclusion column. And in the case of some of the cases of the analysis of our samples, there are some interactions of our samples with the column that are not so much desired, which can be either electrostatic interactions in the case of the polymers that are charged, and hydrophobic interactions if we have extremely hydrophobic samples. So we can see here the example of polar organic solvents, where in the case of the DMSO, if we use the DMSO without the addition of the salts, we can see the splitting of the peaks on the chromatogram, which suggests usually the interaction of the sample with the column. And just by adding a 5 millimolar of the sodium nitrate, that peak can be more symmetrical and we can avoid this electrostatic interaction. Apart from the sodium nitrate, we can use also other salts such as lithium bromide that is mostly used as an addition in the DMF to avoid these ionic interactions. When it comes to the aqueous systems, of course, when we choose which buffer exactly we are going to use for our samples, we need to think about the solubility of our sample. And the second, we need to think about the sample type that we are analyzing, because in the case of the aqueous system, we can have a neutral polymers that are, let's say, the most simple samples that you could have, because you can go with as simple as 0.1 molar of sodium nitrate and start the analysis from there. But when it comes to the polymers that can have either a negative charge or a positive charge, such as polyglutamic acid or polylysine, you need to think about the pH of your buffer and the addition of salts in order to prevent these ionic interactions. And when it comes to as said, there are some samples that are even more complicated, and you can have either proteins or peptides or even the polymers, the block polymers that can have both the positive and the negative charge. And in that case, it's not only the type of the salts, the salt that you are using for your mobile phase, but it's also the concentration really important, and the concrete characteristics of your sample. Because in the case of the hydrophilic polymers, you would need to use high ionic strength, while in the case of the hydrophobic, the lower concentration of salts, lower ionic strength, will be much better choice for your samples. Here you can see the concrete example and how the chromatogram look like in the case of hydrophobic polymers and hydrophobic samples. If we increase from bottom to the top the ionic strength, we can see how the peak changes, meaning that it starts first on the second and the third chromatogram from the bottom. We can see that it starts tailing, and then at the top there it is also split in two, suggesting the interactions of your sample with the column. While in the case of hydrophilic samples, by increasing the ionic strength, we will prevent this interaction of the sample with the column, and going from the peaks in the first, from the bottom to the top, that you are losing the tailing on the right side and you are getting a more symmetric peak and a lower retention time, since we are preventing this interaction of the sample with the column. So now that you know how to choose the mobile phase for your analysis, we are going to move to the next important part as well, which is the choice of the column. When choosing the column, it is also important that you already know which solvent you are going to use for your analysis, because that will influence on your column selection as well. As you can see here on this graph, there are different type of columns that can be used, and most of them are separated according to their stability in the water, aqueous solutions, or the polar organic solvents, or the typical organic, more hydrophobic non-polar organic solvents. So depending on that and your sample, you will choose appropriate column, depending if you are using the aqueous or organic solvent. But apart from that, there are also different strategies when it comes to the column selections. And in most cases right now at the market, we have a different type of columns. Generally, the columns are divided according to the molecular weight separation range that they can be used, because we can have the pores that are larger, medium, or a small one, and they will affect the molecular weight separation range. Or we can have a mixed bed packing columns where they can have all three of these pores inside the column to have a bigger range of the molecular weight separation, or the newest strategy where you can have a multiple pore size, meaning that you can have all of them at the single bead. And depending on your purpose and how big molecular weight the distribution is of your sample, you can choose the column according to that. One of the examples that I would like to show you today is that apart from having one column in your analysis, you can also have a column set where you can combine a different type of columns to have a much better separation of your samples. And in this case, when receiving a sample, we also get a suggestion of which type of column can we use for a specific sample. And as you can see in here, we obtained the 3 different columns in the series that we needed to analyze our samples. However, when analysis was performed, we could see that in this case, the sample was not enough separated from the mobile peak and the salt peak, but also we could see that the left side of the chromatogram was actually pretty empty, which means that we do not maybe need such a high molecular weight range, meaning that in the next step, what I did was removing the high molecular weight column, and instead of that one, I added an additional one, which is at the end, which covered a smaller molecular weight. When analyzing the sample with this set of columns, we could see that there was a better, but still not good enough separation. And again, there was a part of the chromatogram showing that there was no need for a high molecular weight range. And again, in this case, we removed now the 5,000, the higher molecular weight column, and we added the lowest possible column, which showed, as you can see in this yellow circle, acceptable separation of our sample with the salt peaks and as well the mobile phase peaks. So as you can see here, both the eluent and the column are combined together and depends from one another. And the next step that we need to think about is the use of the detectors for our size exclusion experiments. And the use of a detector will most of the time depend on the information that you will need. And that information, when it comes to the polymers, it is a very complex, and the molecular information that you can get from the polymers depends on their use. And one of the first basic information is the molar mass distribution, which can be obtained either by using the concentration detectors, such as refractive index detector and UV detector, which I already explained at one of the beginnings. And this method is the conventional method, where you calibrate your column and with the appropriate standards, and then according to the calibration curve, you can calculate the molecular mass. However, you can also use the light scattering detector in a combination with some concentration detector to also get this information, which is more accurate. The second parameter is the architecture, and to get to this information, you will need to add, apart from the light scattering and the concentration detectors such as refractive index, also a viscometer. When it comes to branching, you also will need the viscometer and the light scattering detector in the combination with the concentration detector. And last but not least, when it comes to size, there are two different types of size, which is the radius of gyration and the radius of hydration. If you need the radius of gyration, you will need to use the light scattering MALS detector, while when it comes to the radius of hydration, you will need to use the viscometer. When it comes to the fit-for-purpose data analysis, we will talk about, first of all, of fit-for-purpose molecular weight determination. And for this example, I chose polyethylene glycol and polyglutamic acid polymer, where if we look at our sample PEG-PGA polymer, one of the first approaches to calculate the molecular weight is by using the conventional calibration, where we can, of course, choose the polyethylene glycol standard, calibrate our column, and calculate the molecular weight by calibration curve. However, if you go a step back and if you look more precisely onto the structure of the PEG standard and the PEG-PGA polymer sample that you need to analyze, they can have the same molecular weight, such as in this example, 10 kilodaltons. However, by adding the PGA part on the PEG polymer, the size of the polymer can be higher, meaning that the higher size of the PEG-PGA will elute earlier than the PEG standard, and meaning that we will get to the overestimated molecular weight of our sample. So what should we do right now? Well, in this case, or in any case where you need to analyze your sample and you do not have an appropriate standard for conventional calibration, you need to use the more advanced detections such as light scattering. Why light scattering? Well, because light scattering does not depend on the elution profile of your sample and the standards, but it depends on the intensity of light that is scattered. And exactly in this case, we combined and compared both and the methods for the molecular weight determination, the conventional calibration and the light scattering one. And as you can see, the conventional calibration overestimated both the molecular weight and polydispersity. And is from a pure chemistry point of view, you could see that and say that this 2 kilodalton is not a significant difference. However, if we think about the use of PEG-PGA as a drug delivery carrier, if you inject these two polymers of the 10 kilodaltons and the 7 kilodaltons, the 2 kilodaltons or 3 kilodaltons difference, we can see a significant difference of the biodistribution between the organs, but also inside an organ. So an accurate molecular weight determination is extremely important in this case. We also need to think about the architecture of the standard NR sample. For example, if we have the pullulan, which is a linear polymer and has a 400 kilodaltons, and the dextran, which is a branched polymer and has the same similar molecular weight, 410 kilodaltons, they could have, let's say, similar molecular weight. However, as you can see here on this chromatogram, they have a different dilution time and suggesting that they have also a different size, meaning that the pullulan as a linear polymer, we will not be able to use it for molecular weight determination of branched polymers. And the vice versa. Dextran, it's only you can be used for the branched polymers and not for the linear one. We can also think from the other point of view. In the case of these three groups of polymers, the linear one, the star, and the cross-linked ones, they can have the same size, right? However, they can have a completely different molecular weight, meaning that all these three groups of polymers, they can elute at the same time, however, they can have a different molecular weight, which means that to actually check the molecular weight of the accurate molecular weight of these samples, we need to use advanced detection such as light scattering. When it comes to the structural analysis, one of the biggest project that we are currently working on is the cross-linking of the star-shaped polyglutamic acid, that in order for us to increase the size of this polymer that will be used as a drug carrier, meaning that by increasing the size, we can have a more tumor accumulation. Well, to characterize this success of the genopin cross-linking of this star PGA carrier, it is a really complex analysis. And for me, it is not just enough to use the DLS or the refractive index, for example, detector, where in this case on the chromatogram, we could see the abundant peak that corresponds to star PGA carrier and the little shoulder and the small peak that could suggest a cross-linking. However, if we look at the light scattering detector, we could see that there is an abundant peak for cross-linked particles because they have a higher molecular weight, and a small peak that corresponds to a star PGA carrier that is not cross-linked. So instead of using one or another, we suggest the simultaneous multi-detection platform use, both the refractive index and the light scattering, in order to much better elucidate this fate of the star PGA carrier cross-linked one in the biological conditions. However, we didn't want to stop there. We went even a step further. And we also need to know about the shape analysis and the shape of our polymers, these cross-linked polymers, where apart from having the data like a radius of gyration and the molecular weight, we also need the radius of hydration. And why do we need to know something about the shape analysis? Well, it's the same story as for the molecular weight, because the shape of the cross-linked polymer will influence the biodistribution of your material. And how are we going to do that? How are we going to have this shape analysis? Well, Let us see in here on the example of dextran with a high molecular weight. In the green line, you can see the molecular weight distribution that we obtained by the light scattering. And the first thing that we can notice is that there are two different slopes in the green curve. And that the higher slope on the higher molecular weight distribution is not possible to be detected by the column calibration method, but only by the light scattering. And then second, you can see two additional curves that suggest the distribution of the radius of hydration, the pink one, and the blue one, radius of gyration, the distribution of the radius of gyration. And as you can see, these two curves have a different slope, meaning that this ratio between the radius of gyration and radius of hydration can suggest us about the structural shape and the conformation of the polymer inside the solution. In the case of the low molecular weight, we can see that the radius of gyration is higher than the radius of hydration, suggesting a random coil structure, while in the case of the high molecular weight region, dextran has a higher radius of hydration than the radius of gyration, suggesting that we have a more dense structure, which is indicated mostly for branched polymers. So we used this structural analysis for the analysis and the characterization of HPMA polymer that is used as well as a drug carrier. And the first thing that we noticed when analyzing the ratio between radius of gyration and radius of hydration is that the increase of the molecular weight of the PHPMA polymer can lead to a change of the conformation from random coil to a compact one, but as well, once adding the drug and conjugating the drug to this polymer, we can extend the structure, going from compact into the extended rod of our polymer. So as you can see so far, I've shown you a lot of examples of the data that we obtained from the SEC, coupled with a different advanced detection. And everything is great. You can get a lot of information from this technique. But one of the things that we all need to think about and we need to talk is the 3R in science, which stands for robust, reproducible, and repeatable. These are the three most common, overused, and often misused attributes that are used either for describing some biological or a chemical process. But in order to have all of these robust, reproducible, and repeatable, the 3R, we also need a 4R, the fourth one in science, which is related to the reliable instruments. So how are we going to get to these reliable instruments? Well, in the case of the GPC, the first thing that we need to consider is the temperature control of our GPC system, meaning that all of these instrument setup that I explained at the beginning of this presentation should be in a full temperature control system. In the case of the system where there is no temperature control, you can see here on the left graph the pink line that suggests the change of the elution time of the samples throughout the sequence and the number of rejection up to 100. That change corresponds to the change of the room temperature. So this is the system if you will have every component of the SEC system, GPC system, separately and not with the full temperature control. All of these can influence your precise molecular weight determination. However, on the right side, on the graph on the right side, we can see the results of the system with the full temperature control, where the elution time remains the same no matter how the room temperature changes throughout these injections. And the second thing, what makes your instrument reliable is the reliable detector, such as in the case of the refractive index. And there are currently two different designs of the refractive index. One, the conventional one, where you can have a reference cell that is containing the stagnant fluid, stagnant reference fluid, and during the time of the analysis and the sequence, this solvent, in the case of the organic, can be degraded, or in the case of the aqueous, you can have a deposition of the salts, meaning that you will have a shift in the baseline levels of the refractive index, as you can see here on the graph, throughout the 200 minutes. And all of that can influence on the accuracy of your measurement and the molecular weight determination. While in the case of the dual flow refractive index cell, the reference cell contains the flowing solvent, which means that the baseline of the refractive index detector is much stable, and you can get a more repeatable results. As an example of this case, we can see here the analysis of 8 replicates of high molecular weight hyaluronic acid, where you can see the repeatability of the signal, both refractive index and light scattering. However, as well, don't trust just the images. You also will need to ask for the data. And if we see the data in here and the analysis of the molecular weight, first thing that we can notice is the coefficient of variation that we calculated for the replicates, 3 replicates, and it varies from 0.06 to 0.08. These values shows a really low variability of the instrument, meaning that we have a really good repeatability of our instruments. When we move from these values to the calculation of the coefficient of variation from all these 8 replicates, we increase the coefficient of variation to 0.67, meaning that how repeatable I am personally when preparing the samples influences by 0.6% to an instrument, to the experimental error, which is not so bad for a human, right? All of that leads us actually to the conclusion that there is one thing to have the robust, reproducible, repeatable data that you are getting from the reliable instruments. But in order to have the 4R in science, you will also need good scientists, which will not only make sure to have the 4R in science, but will also take a good care of these instruments to have these 4Rs. So with that, I'm getting close to an end of this presentation, and I really hope that I managed to transfer you how the GPC and can be used together with advanced detection as a method of choice for the characterization of polymers. And I know that they just moved through these couple of parameters that can be used for the characterization of drug delivery system. However, we are also currently exploring the use of this technique for other physicochemical parameters. So stay tuned to get the new results. And at the end, I would just like for you to take home the key messages, such as the use of light scattering for direct molecular weight and size determination. Then the use of light scattering and viscometer for structural analysis, meaning that apart from molecular weight, RG, you can also get RH. And of course, make sure that you have the reliable instruments and the scientists, because only by having all these 3 components into account, you will make sure to have the robust, repeatable, and reproducible data of your studies, which will then in turn give us much better therapies for patients in clinic.